Good morning. We were seeing question number 61, JPSC JET, Life Science detailed video. So, question number 61 says agrobacteria mediated transformation involves transfer of which DNA region? The options were tDNA, ORIC, operon DNA, cosmates. Isn't it? This is the question. So, we will see each of the terms involved in it. So, let's begin. Whenever talking about a cell, your cell image is something like this, isn't it? So if we complex a bit and visually make it beautiful, and so the cell will look like this, isn't it? So interestingly, all the organism have, whether it is a plant or an animal, they are made up of many cells, and every cell demonstrate the central dogma of life. Every cell, which is every organism which has its own DNA, is due to presence of a certain information present inside the DNA, isn't it? And that information is passed on from one generation to the next generation. So the central dogma of life, from DNA the replication takes place, and from DNA the mRNA is transcribed, and mRNA is further processed to make the protein which we see from our naked eyes. Here again, the central dogma of life, DNA is transcribed into mRNA and it is then translated to form protein, isn't it? So when you talk about a chromosome, the chromosome is like a book and the DNA is like the pages of the book, isn't it? And the chromosome is the compact part. These are all the figures which demonstrate what a DNA and chromosome are. So in humans, 46 chromosomes are present, isn't it? 23 pairs, and that because of that information, humans is visualized as humans, and other organisms are have different phenotype or different look. An elephant is an elephant due to the information present in its chromosome number or its DNA, isn't it? Here are some few animals and their number of pairs of chromosome, isn't it? So the number of pair of chromosome has nothing to do whether with the size of the organism, isn't it? So the point is, the DNA is responsible for the phenotype or how the organism look. And now the question arises, what if we change the DNA? Will the organism change? And can we do that? Is it possible that if we change the DNA in an organism, the number, the phenotype changes, or the looks, how the organism look, it changes? And yes, that is possible, and that is known as biotechnology, isn't it? So the thing is, biotechnology is simple in single cells, and with the complexity of an multicellular, and the number with the increase in number of cells, the complexity increases. So single cell like bacteria shows this biotechnological property, and from bacteria we have learned about this field, and we have given this biotechnology field a foundation on, and its principle have been founded on the basis of how the agrobacterium works, isn't it? So agrobacterium tumefaciens is a soil bacteria. See, in case of bacteria, there are two chromosomes. The main chromosome is known as bacterial DNA, and the extra chromosome is, which is a circular chromosome, is known as plasmid. In agrobacterium tumefaciens this is a rod-shaped bacteria, gram-negative, present in the soil. It also has two chromosomes. One is bacterial chromosome, and one is an extra chromosome that is known as circular chromosome, that is known as plasmid, isn't it? In case of agrobacterium, it is known as Ti plasmid. And how this is related to the plant, we will see about that. See, what an agrobacterium does, it infects the plant and causes crown gall disease, isn't it? So the name of that T plasmid is Ti plasmid because it is shortened for tumor-inducing plasmid, isn't it? So when the agrobacterium infects the plant, it gets food from the plant, and in return, the plant hosts the agrobacterium for its hormonals or hormone or another hormone like cytokinin or auxin. See, let's see in detail. Okay, whenever a tDNA or an agrobacterium finds a wounded site on the plant, it infects the plant and it causes a crown gall disease, and it sends its tDNA, that is the part of the Ti plasmid, to the plant, and the tDNA incorporates inside the genome of the plant and makes the food for itself. So here is the Ti plasmid details, isn't it? There are 4 regions: tDNA region, isn't it? In tDNA region there is the regions between right border and left border, as you can see. You can also see here the Ti plasmid consists of 4 parts, isn't it? 4 regions it consists. Region A is tDNA, which is infectious, and region B is opine catabolism gene. Opine is being catabolized, which is obtained from the plant. Region C is origin of replication, and region D is virulence gene, which is responsible for Vir protein, isn't it? So we will see this process. We will see this step by step. Okay, let's begin, and this is the figure we will see as a reference. Okay, see the at 1A, specific attachment of the plants takes place to the bacterium. The bacteria attaches itself to the plant cell, which is wounded, and the bacterium senses the plant derived signal which is secreted by the plant cell, that is acetylserinone, and then the plant derived signals are recognized by Vir A protein. Vir A protein is secreted by virulence region, isn't it? And due to this virulence region, Vir G gets activated, isn't it? Vir G is nothing but transcription. Vir G causes the transcription of another virulence gene, Vir B, Vir G, Vir C, Vir T, E and F and so on. These Vir proteins, Vir D2 protein gets attached to the tDNA, isn't it? tDNA is a part of Ti plasmid, as we have just discussed, and that Vir D2 takes the tDNA inside the plant cell, and Vir D2 is protected by Vir E2. Okay, because why the protection is needed? Because without protection of Vir E2, the plant cell and the plant defense system will degrade the tDNA, and then again Vir D2 takes the DNA inside the nucleus, in which the tDNA attaches itself to the plant genome, isn't it? So let's see this again step by step. Okay, so first, what is done when the agrobacterium identifies an wounded plant cell, isn't it? So the bacterium attaches itself, and the recognition is done by the wound recognition is done by Vir A. Vir A is the sensor histidine kinase, and this is activated by acetylserinone on phenolics, this is the compound that the plant secretes when it is wounded. Okay, so acetylserinone or phenolics is secreted by the wounded plant cell, isn't it? And the Vir A recognizes the acetylserinone or the phenolics. So when this Vir A detects this acetylserinone, it activates Vir G, isn't it? Then what the Vir G takes place due to the phosphorylation of Vir G again acts as a transcription factor, isn't it? And transcription factor, and it activates, it transcribes the other virulence genes D, virulence B, E1, F, and so on, isn't it? So again, vir D2 acts as endonuclease and cuts DNA, tDNA. Okay, and T2 attaches to the tDNA itself, isn't it? And helps, and D1 helps D2. So now D2 comes and cuts the tDNA, isn't it? And the N gets attached to the tDNA. Okay, now after, again, when Vir G was phosphorylated, and when Vir G acted as transcription factor, other Vir proteins were also transcribed. And again, Vir D2 acted as endonuclease, and it has cut tDNA and has attached to the tDNA. Okay, and other Vir protein, that is D4 and D11, forms T4SS type secretory system. This has nothing but a fancy name for a channel. Okay, the virulence protein, uh, specifically Vir D4 and D11, makes the channel, isn't it? So through which the tDNA is being transferred. Again, Vir C1 and C2 binds to the overdrive region. We will see what overdrive region is, and what it does. It increases the synthesis of tDNA, and Vir C1 C2 also gets pushes the Vir D2 and tDNA towards the poles of the agrobacterium, isn't it? Then after, what happens, Vir B, D4 and D11 makes the channel, isn't it, which has a name of T4SS secretion export, and through that channel the T2 DNA, the tDNA is being transported by the D2, isn't it? And with that, E1 also, E1, E2 and F also travels towards the plant cell, isn't it? And T4SS is nothing but the channel through which the tDNA will pass. Now, what takes place? What travels? What carries the tDNA? Where D2 carries the tDNA inside the plant cell. Once the tDNA reaches inside the plant cell, where E2 act as single SSB binding protein, isn't it? Single strand binding protein. So it is so that the plant does not disintegrates or does not nuclease. The nucleus in the plant cell does not disintegrates the tDNA. Okay, so the E2 protects that single strand tDNA, and E1 protects E2, so that the plant proteases does not disintegrates the tDNA. Okay, so the plant defense system is VIP1. Okay, the E1 and E2 protects the tDNA from VIP. Okay, again, now when this is all, Vir F is also transported through that channel inside the plant cell. And what when the Vir F reaches that this complex in which Vir D2 is attached to the tDNA, what it does, it degrades the Vir E2 and Vir D2, releasing the tDNA, and Vir, what that VIP does, it carries the tDNA inside the plant genome and makes it integrates into the host chromosome. Now that the host chromosome has the tDNA of the agrobacterium, what it does is it starts synthesizing opine, auxin and cytokinin. Why will it synthesize opine, auxin and cytokinin? Because it contains, here you can see the tDNA region contains genes of auxin, cytokinin for the plant and opine for the bacterium, isn't it? So this is the summary. If you can go through the summary. So we come upon what the question was. The question was agrobacterium mediated transformation involves transfer of which DNA region. So now you have seen that the tDNA is being transported from the agrobacterium to the plant cell. Option B was Oric. Oric is nothing but the origin of replication in the tDNA Ti plasmid, isn't it? So the origin of replication is not being transported. Option C is operon DNA. Operon is just a part of DNA and not is not transported toward in the plant cell, isn't it? So operon is present in the virulence region, isn't it? So the virulence region, many operon, viridi operon is present, vir E operon is present, and these are not transported, isn't it? And cosmid is a plasmid in which the bacteriophage part is there, isn't it? The cosmid is a hybrid laboratory tool, that is plasmid, used in the genetic engineering to clone and transfer large segments of DNA into bacteria. So what does the cosmid contains? The cosmid contains the plasmid, and it also contains the bacteriophage part, isn't it? So now that you have know that the correct option is tDNA, isn't it? Thank you for having time.